Welcome, everyone, to this week's edition of the Extracellular Vesicle Club. I'm very happy that today this session is being hosted by EViNS, the EVs in Nervous Systems Working Group. This is a special interest group of ICEV, and in fact, it has several working groups within it. And so we're going to hear about one of those working groups today. I'd first of all, though, like to apologize because the email that I sent out contained the wrong link to the paper. It was actually to the paper from last week, which was an amazing paper too. But I will put the correct link here in the chat box, and we'll make sure that that's also linked on the YouTube video later. So I apologize for that. And I'd also like to ask everybody to please, during the presentation, put your comments and questions in the chat box, and then our moderator today will call on you at the end of the presentation. So I'd like to introduce our moderator, Julien Saint-Pol, who has also helped with picking out this paper, which I think is going to be very fun to discuss. Julien, you're part of one of the working groups of EViNS, and I'd like to invite you to take over the screen here and tell us a little bit about that.
Okay, so hello everyone. That's a great pleasure to introduce one of the working group of the EViNS. So the working group number 1 dedicated to EVs and brain barriers in general. We dedicated this group to all the brain barriers, not only the blood-brain barrier, the blood CSF barrier, because there is just so much barrier protecting the brain and the central nervous system. So we would like to introduce this group today with a nice paper. We will be presented by Hermine. This EViNS is constituted by 4 working groups. The first one is dedicated to brain barriers. So I just would like to invite you to join us if you are interested in. The only condition is just to be an ICEV member. Otherwise, you are much more than welcome to join us. So thanks to that, and I also would like to thank my co-facilitator for this group, Rosemary van den Broeke, and also Professor Jason Howitt from Australia. He's not on that slide, but he's just sharing this responsibility with us. With this consideration in mind, it's just my great pleasure to introduce Hermine Counil. She is a young PhD student working in the Charles Ramassami's lab in Laval University in Canada. And she will present a very nice paper sorted out in January in JEV concerning peripheral EVs, their capacity to enter the brain and also to stimulate microglia population. So, Hermine, that's a great pleasure to see you today. And now the floor is yours.
Thank you so much. And again, thank you so much for the invitation. So as you said, I was working under the supervision of Professor Charles Ramassami during my PhD. And so I just finished it a couple of weeks ago, but I'm very happy to present to you the results of my PhD, which has been published, as you said, earlier this year. But also I wanted to, if I can give you some other preliminary results that we obtained following this first article in line of this subject about the penetration of peripheral EVs through the BBB and then interaction with brain cells. So I'm gonna give you like a very academic presentation with a first quick introduction of why we worked on this hypothesis and area of research. So as you may know, Alzheimer's disease, or AD, is an age-related and incurable neurodegenerative pathology, which affects mostly adult people. It is progressive neurodegenerative disease, which leads to cognitive impairment, memory loss, and mood disorders. And currently, it is diagnosed 10 to 20 years after its beginning, after the onset of the symptoms, because the mechanisms are not very well understood. So currently, the prevalence of the disease is about 1 million of people, and it is expected to increase in the next years. So it is why AD is a very important disease to study nowadays, because of its prevalence and the fact that there is no curable treatment currently. So AD has been well characterized for the abnormal extracellular accumulation of the peptide amyloid beta in neurons, around neurons, but also about the intracellular accumulation of abnormally hyperphosphorylated tau proteins. So those two leads to neuronal dysfunction. but also to neuroinflammation with notably the activation of microglia cells and astrocytes in the brain. Progressively, it leads to the disruption of the blood-brain barrier integrity, and finally to neurodegeneration. And so together, this leads to the cerebral atrophy, which is characteristic of Alzheimer's. And notably, we have a very well-specific atrophy of the hippocampus, which is the primary area for memory storage. So even if we don't really understand all the mechanism behind Alzheimer's disease, we know some of the risk factors, such as genetic, for sure, but also inflammatory diseases. So it has been demonstrated that the alteration of the immune system, as well as the metabolic diseases and other systemic inflammation, such as virus infection, can increase the risk for Alzheimer's pathogenesis. So it is known that these chronic inflammatory diseases are responsible for the liberation, release of chronic inflammatory cytokines and factors such as IL-6 or TNF-alpha. And so those tissues release in the periphery and notably in the blood toxic compounds on a chronic way. And thus, they may affect the brain homeostasis and could lead to Alzheimer's disease, but the mechanisms are not very well understood. So this is why we were interested in EVs, because EVs, as you may know, are nanoparticles released by our cells. They are non-replicable, but are composed of very varied and functional cargo, which depends on the state of the donor cells. And these EVs can be classified into different subpopulations according to the synthesis pathway, the size, or also their origin. They have, because of their characteristic properties, different role and can have different organ tropism and be internalized by specific cells according to different pathways. We know that EVs have a role in intracellular communication, and they can influence receptor cells through physiological or pathological pathways, depending on their composition. And so they can influence receptor cells through either autocrine, paracrine, or endocrine pathways. And also they can reach the different biofluids of the body, such as the blood. And so they could circulate into the whole body and target very distant organs. In the brain, it has been demonstrated that brain-derived EVs contribute to brain homeostasis. So all cell types, such as microglia, astrocytes, neurons, or also the BBB endothelial cells, for example, can produce and release EVs. And these EVs participate to the homeostasis through different mechanisms, such as phagocytic activity, synaptic plasticity or transmission, but also controlled inflammation. So in an healthy condition, they have homeostatic function. And in AD, it has been demonstrated that brain-derived EVs are enriched in amyloid beta to hyperphosphorylated and other inflammatory factors, such as cytokines. But these compositions have been, like the role of EVs in AD has been shown first to be able to preserve the brain functions, such as the synaptic activity, because EVs can secrete some toxic compounds, such as oligomeric beta. And also they can deliver these toxic compounds to glial cells, such as microglia, which then degrades them and avoid any contamination, if I can say, of the brain. In addition, it has been observed that peripheral EVs, that we could here call PEVs, which can be found in the blood of patients, are enriched in some biomarkers for AD when the patient has the disease. And so it suggests that EVs coming from the brain and then going to the blood circulation could have a role in the elimination of the toxic compounds from the brain through peripheral organs, such as the liver. But in another way, EVs, brain-derived EVs, have been observed to have pathologic functions and that can contribute to Alzheimer's disease pathogenesis. So, for example, EVs enriched in amyloid beta or tau phosphorylated has been observed to induce synaptic dysfunctions through different molecular pathways and cellular pathways, either inside the neurons or at the outside of the neuron, and then they can propagate through the interconnected neurons and then could participate to AD pathogenesis. In addition, in EVs, which has been released by microglial cells, have been observed to contain a high level of TNF-alpha and IL-6, which has both pro-inflammatory cytokines. They can trigger the polarization of the microglia from an homeostatic state to a pro-inflammatory state. They also promote the activation of inflammatory pathways in neurons and link them to synaptic and neuronal dysfunctions. And in addition, it has been observed that EVs, brain-derived EVs from AD patients induce different type of alteration related to Alzheimer's and cognitive dysfunction in mouse model when injected in the brain of the animals. So here we were interested about EVs from the periphery and not from the brain, because as I explained it earlier, we know that metabolic diseases and chronic inflammatory pathology are risk factors for AD, but we don't really know what is the link between these inflammatory factors and Alzheimer's and the brain. So we had this idea that EVs could contribute to the communication between the periphery and the brain. So we know that brain-derived EVs can be detected in the blood. Others and our laboratories have already published about that. But the passage of peripheral EVs from the blood to the brain is, well, not very well understood. Some studies have demonstrated that EVs from an healthy condition can cross the BBB when only the BBB is affected by an inflammatory state, such as TNF-alpha treatment in vitro, while others have demonstrated that pathological EVs can cross the intact BBB and then be internalized in the brain and lead to brain dysfunction, notably in mouse model. But so the question here was first, do peripheral EVs can cross the blood-brain barrier? And so we had this hypothesis that yes, they can cross the BBB, and then they're internalized by neuronal and glial cells. And so this would lead to neuronal alteration and neuroinflammation, which would be associated with AD etiology. So we had three main objectives. So the first one was to isolate and characterize peripheral EVs from the blood of either healthy adults or connectively healthy adult patients or Alzheimer's subjects. The second one was to evaluate the passage of the vesicles of peripheral EVs across the intact BBB, in vitro and in vivo, and then to confirm their internalization by brain cells. And finally, to analyze how peripheral EVs may influence neuronal and human cells activity. So first, we did the isolation and characterization. So here, I just wanted to present you the first article, but also other results that we obtained following this first publication. So for the article published, we used our first technique, which is based on the isolation of the vesicle from the plasma of adults, EDOLI, and mild Alzheimer's patients. And we used a precipitation technique based on the PEG. And then for the second technique, which is currently in preparation for the article, we use a sera, not the plasma, but the sera of either EDOLI or mild Alzheimer's patients. And we did this isolation by using the SEC technique. So I'm going to show you the result we obtained with both techniques just to have an idea and compare. So we have here team images of the samples following isolation. So we can see that we have an enrichment, a small particle with some likely to exosomes. And with NTA, we demonstrated that we have, with both the technique, an enrichment of small vesicles between 50 and 250 nanometers of diameter. What was very interesting here is that we observed that we have a certain concentration of the vesicles for adult patients, but with healthy aging, we have a huge decrease of the concentration of the small vesicles between 50 and 150 nanometers. While for AD patients, from both plasma and serum, we have a huge, significant increase of the concentration of the small vesicles. And it seems to be very specific to small vesicles because after up to 150 nanometers, we don't have the same results. So it's very specific to 50 to 150 nanometers. So it seems to be independent on both biological samples, but also the isolation technique. In addition, we did a characterization about the protein contents, like some of the proteins. So we didn't have significant difference between the groups about the protein concentration. It was very similar. And we have also an enrichment observed by either classical Western blots or capillary Western blots of some proteins involved in vesicles biogenesis. So it was a good way to confirm that we have an enrichment on vesicles. And so here I have new data that we obtained in the past few months about the samples isolated from the sera of patient by SEC. So we wanted to measure the level of cytokines, some cytokine pro-inflammatory and anti-inflammatory contained into vesicles from EDOLI or AD patients. We did MSD analysis to measure these cytokines, but also to have some idea of the concentration contained in the vesicles about A beta and tau. So surprisingly, we didn't have any difference about these cytokines between the two groups, neither for A beta, even if we have like variables depending on the patients. And tau proteins, and its phosphorylated form 231 were not detected. So it's just an idea to keep in mind for the next result I'm going to show you later, because it's going to be important to understand what is happening once in the brain. And finally, what was very important for us was the leveling of the vesicles, because we wanted to follow their biodistribution in the different models that we used in vivo and in vitro. So we used the PKH and exoglow fluorescent probe. So we know that these fluorescent probes are like questionable for some. Totally agree with that, but it was in our condition hard to have another probe because we were working on human samples and vesicle coming from all different part of the body because they were in the blood. So we used this fluorescent probe and did some quality control analysis by using nanocytometry to confirm that we have an enrichment like a fluorescent EVs and no lipidic particles, for example, contamination. And if we had some contamination, we didn't use our samples. So just to summarize a little bit this first objective, by using two different techniques, two different biological samples, serum, plasma from humans, we observed that we have a decrease of the concentration of vesicles with LC gene, but the vesicles number of small particles significantly increase in mild Alzheimer's patients. So then we wanted to evaluate if the vesicles could cross the BBB, the intact BBB, and then be internalized by neuronal cells. So first, we used a BBB in vitro model based on a transwell. So we have endothelial cells from a murine model to mimic the BBB, and then in the lower chamber, we have either SKNSH human neuronal cells or HMC3 human microglial cells. So we did different type of techniques to characterize this model to be sure that it was working well and made sure that the BBB was correct. And then we treated this BBB model with our fluorescent EVs. So here you have the result 24-hour post-incubation between the endothelial cells, so with the nucleus here in blue, the one tight junction protein in green, and then in red, you have the vesicles. So either here from EDOLI patients or here from Alzheimer's patients. And so we can see here what we have colocalization between the vesicles and the endothelial cells. And by using 3D confocal imaging, we were able to see that we had vesicles both on the surface, but also inside the endothelial cells. We confirmed that we have an interaction between the vesicles and the endothelial cells by using flow cytometry. So interestingly, we didn't have a difference of level of internalization, colocalization between the vesicles and the endothelial cells independently of the group. And also, interestingly here, we quantified a certain amount of the vesicles into the blood-phase chamber, if I can say, the luminal compartment, but also into the abdominal compartment, which would be the brain phase. So it seemed that the vesicles are internalized by the endothelial cells, and some of them are then released in the lower chamber. So we wanted to see if these vesicles could affect the integrity of the BBB, which could explain why we have a passage for some reason. So we did different characterization, and what was interesting is that the tight junction proteins, claudin 5 and zonulin 1 were not alterated following the treatment with the vesicles, but following incubation with EVs from 80 patients, we have a significant decrease of the transmedullary electrical resistance of the endothelial cells, which suggests that the vesicles from 80 patients may impair the BBB sealing on a long-term treatment. So we didn't went in deeper analysis here, but it would be interesting to see, especially in animals models, what would happen on a long-term incubation, like treatment with a vesicle, what happened to the BBB. And to confirm these results that we obtained in vitro, we wanted to continue the follow-up of the biodistribution and interaction with the vesicles and the cells in vivo. So to do that, we used a zebrafish model at the larvae stage. It's a very nice model because you can follow up in real time how the vesicles are interacting with the cells. So we use a first transgenic model, which expresses the endothelial cells in green, as you can see here. So here, this is the focus on the head. You have the eyes here. The brain is up right here, you can imagine it. And in red, you have the vesicles injected in the blood circulation. So either 1 hour or 24-hour post-injection. So we observed that we had global distribution of the vesicles into the body, but with the time, we have an increase of the fluorescence intensity in the head compared to the whole body and not in other parts of the animal. So we were very, very interested of what was happening in the head. So we did focus on the head, and by using the iMaris software, we were able to do a 3D reconstruction of our zebrafish shells. So you have still here in grid the vessels, the BBB, and then in white, you have the area of colocalization between endothelial cells and the vesicles in red. So this model was very nice because, as I said before, we were able to see in real time what was happening. So here you have the blood vessels around the eye of the zebrafish. The head is here. You can see heat in the black area. This is 1 hour post-injection, and you can see the white dots here are the area of colocalization between the vesicles and the endothelial cells. So the vesicles are going from the blood to the brain through their passage with the endothelial cells. So we then wanted to know what was happening after their passage through the brain, because it could be degraded or just moving around the extracellular space or be internalized by cells. So we used the two models that we had previously. So the transmod model in vitro and the zebrafish model in vivo. So in vitro, by using still the transmod model, we confirmed that we have an internalization of the vesicles by the human microglial cells. So here you have confocal imaging of the vesicles in red and the cells in blue for the nucleus, and green for marker for SK-message cell. And we confirmed that we have indeed an internalization of the vesicles following 24-hour incubation into the BBB model. So the vesicles are able to pass through the BBB and then be internalized by neuronal cells. And in vivo, we observed similar results, meaning that following their injection in the blood circulation, the vesicles were able to go into the brain and then being internalized by neurons. So you have here in green the neurons, and in white all the area of colocalization between vesicles and neurons. And here we didn't have any difference between the group for the intensity of colocalization between the cells, neuronal cells, and the vesicles. So still here, a quick video to show you what was happening. So you have the blood circulation here. We can see some cells passing into it. You have the neurons in green, and then you have the interaction between neurons and vesicles in white. So it's not very easy to see it, but here we have neurons. So this is why we have a lot of white dots in this area. So we can see that we have a lot of vesicles interacting with the neurons close to the blood vessels. And so we wanted also to confirm that with glial cells, because as you know, glial cells are very important for the homeostasis of the brain, and they're involved in the AD pathogenesis. So in vitro, we used HMC3 human microglia cells. So we confirmed that we had an internalization of the vesicles by microglia following a treatment with the BBB. And what was interesting here is that the level of colocalization between HMC3 and the vesicles were different between the three groups, and was also lower compared to neuronal cells. So it seems that the HMC3 cells are highly dynamic and seems to degrade at least the signal related to EVs and probably EVs2 very quickly, which fits where the function of immune homeostasis. Also in vivo, we use another transgenic model for zebrafish to express here in green glial-like cells in fluorescence. So we also injected the vesicles into the blood circulation and observed a certain amount of vesicles colocalizing with glial cells. And interestingly here, we had increased of colocalization between vesicles from elderly patients, 24-hour post-injections, but not for AD patients. So it seemed that here, so depending on the vesicles and their compounds, that the degradation or maybe interaction with the glial-like cells may be different. So here you have 3D reconstruction imaging obtained with iMaris software. So you have in green the glial-like cells, which is nice because you can see very well the blood vessels inside the brain. In red, the vesicles, and I hope you see it well, but in purple you have the colocalization area. So it was very nice to see that we had a lot of colocalization area, even in deep tissues after the passage through the BBB. And one important result that we obtained in this first article is about how microglia HMC3 cells react following treatment with vesicles. So we were interested about the shape, the morphology of the microglia cells following their interaction with the vesicles in the presence of the endothelial cells, the EViNS cells, because we observed that with the transfer model, compared without, EVs from AD patient induced a change of the shape of the microglia cells, which became amyloid, which is characteristic of AD. And so to understand a little bit more what was happening here, we decided to do an ELISA dosage of IL-6 cytokine, which is a pro-inflammatory cytokines involved in AD. We know that we have a huge increase of these cytokines in the brain and also in the blood of the patients. So we had different treatment conditions as positive control for inflammation, such as LPS and amyloid beta. So we observed that indeed this inflammatory condition increased the release of IL-6. And then we treated with the vesicles. So the control vesicles from elder patients didn't induce upregulation of the release of IL-6. But the vesicles from EDELI, and more importantly from Alzheimer's patients, induce a high release of IL-6, which were higher than the pro-inflammatory conditions, LPS and A-beta. And what was very interesting here is that in the presence of the transfer model, we still had this increase of IL-6 release. So of course, in the abdominal phase, which is the brain phase, but also in the liminal phase. And here we used a BBB model based on the EViNS, which is a neuronal cells line. So where we were dosing IL-6 from only human, it proves that IL-6 dosage in the liminal phase was coming from the microglia cells because they were human. So it's interesting to see this type of result because it suggests that maybe the increase of IL-6 that we are able to observe in the blood of patients may be produced by microglia cells in the brain. So to summarize this second objective and results, which has been published already, we observe that we have a decrease of the amount of the vesicles from EDELI patient in the blood during LC aging, but for AD patient, mild AD patient, we have a significant increase. These vesicles are able to cross the intact BBB in vitro and in vivo, and then they are internalized by microglial cells, neurons, and at least induce a change in the activity of microglia, notably with an increase of IL-6 production. So we published these results earlier this year. And then I wanted today, if I still have some time, a few minutes maybe, to present you other results that we obtained in the couple of past few months. So because of the first results that we obtained, we were very interested about how peripheral EVs from EDELI and a mild Alzheimer patient modify the activity of neuronal and human cells. So we continue to work in vitro with in vitro models. So here, this is the results obtained from the sera of patients, EDELI and AD, isolated with a SEC. So we still confirm that we have an interaction between the vesicles and here HMC3 cells. And then still by MSD dosage, we analyze the vesicles either anti or pro-inflammatory cytokines, not the vesicles, the pro and anti-inflammatory cytokines released in the media following treatment, 24-hour treatment of the cells with the vesicles. And so just to summarize in an easier way, maybe we obtained that EVs from EDELI patients induce increase of interferon gamma and IL-6 from microglia cells, while EVs from Alzheimer patient induce an increase of IL-1 beta, interferon gamma and IL-6, but also of IL-4. So it suggests that microglia cells are responding differently and probably adopting different phenotypes according to the vesicles and their composition and origin. And additionally, we did some immunochemistry tests to see how was the cells, notably the morphology, and we observed that we have an increase of the expression of IB1 and CD68, particularly after the incubation with peripheral EVs on AD patients. So it suggests that the cells have a higher activity following AD treatment. Similarly, we did also experiments with neuronal cells in vitro. So we used two different cell lines, one from murine, which overexpressed the amyloid beta precursor proteins. And so this model is able to produce amyloid beta. And so following treatment with the vesicles, we had a significant increase of A-beta released only from AD patients. So if you remember well, the first experimental result I showed you in the M1, we didn't have any difference of A-beta levels between vesicles from EDELI and vesicles from AD patients. So here, the A beta produced is really from the cells. So it means that the vesicles can induce release of toxic compounds, like at least proteins associated to Alzheimer's disease. We also wanted to see what was the effect on the tau protein and its phosphorylated form, TER231. So we used the human NSH cell, and unfortunately, we didn't have any difference between tau proteins and tau phosphorylated between the two groups, but the ratio was decreased by the two types of vesicles. The phosphorylated form and total tau proteins ratio was decreased. So it may suggest that the vesicles could have an impact on the homeostasis of tau and its regulation of phosphorylation. So just to summarize this third objective, we demonstrated that in the blood, we have an increase of vesicles from 80 patients. I didn't show you today, but we also observed that the vesicles have had an effect on monocytes and lymphocyte TC4+ cells in vitro. And then following the crossing through the BBB, they are internalized by glial cells, microglial cells, and neurons, where they are able to induce either the release of pro and anti-inflammatory cytokines or A beta, decrease also the ratio between phosphorylated form 231 and total tau. And also we observed that we have a change of the synaptic activity ex vivo following the treatment with the vesicles. So to conclude all of these results, that first article and the second one that is in preparation strongly suggest that the vesicles may have a role in AD pathogenesis and, globally speaking, in brain homeostasis. So for sure, it is now required to do other experiments in vivo and probably using animals model to confirm that we observed. But it is, I think, a very interesting new pathway to better understand the communication between the periphery and the brain. So with that, I would like to acknowledge my laboratory members, my PI, Charlotte Massami, my colleagues, and my different internship students that worked on this project. Also our collaborators and the different grants that have made this project possible. Thank you. And also, just to let you know that I'm gonna continue postdoc on this area. So if you are interested to do collaboration, or if you have any question, don't hesitate to contact me. Thank you.
Thank you so much, Hermine, for this very great presentation. So basically, I was waiting for your question in the chat box, so it's quite empty. So it's a good opportunity for me to ask the very first one question. So you clearly well demonstrated that the EVs are able to cross the blood-brain barrier, and you didn't show that today, but in your paper, you showed that the temperature could have an impact of this transport across the BBB. So my question is probably the main question that people have. Do you have any clues about the mechanisms of this transport of EVs across the blood-brain barrier?
Yeah, so thank you so much for the question. And indeed, I didn't present it today, but we had a control condition at 4 degrees to see if the vesicles were able to be internalized by the cells. And it seems that no, at 4 degrees, all the interaction were not present, if I can say. So it seemed that the mechanisms are active, but I can't say which one because unfortunately we didn't study this one, but they are still active, and yeah, it's very hard because we didn't study really all the mechanisms in our study.
A second one, all the effects you described with your EVs are quite interesting, particularly for the brain cells, but you mentioned also that you have impact on peripheral cells and particularly for macrophages, if I well followed. Monocytes, sorry. Okay. Did you perform some multi-omics approaches just to be sure to have all the parameters to know why the cells are responding to these EVs?
No, unfortunately, no. But it would be very interesting to go deeper. Yeah, I totally agree with that.
Some question from the chat box. So Susan Golds asks you something dedicated to a technical question. Why did you use PEG with serum and SEC with plasma?
Yeah, thank you for the question. So for the first article, we used PEG because it was already published in our lab. We used it like on a daily process. So I wanted to continue with this technique, but then I wanted also to see how EV is isolated with the different techniques, which are like a little bit different sometimes from a technique to another, would be impacting the cells, the recipient cells. So this is why we choose to use the SEC. And also the SEC is probably more recognized nowadays than the PEG technique.
Another interesting question from Divya Sridharan. Great presentation. Do you know what the percentage of the total EVs were able to cross the BBB?
Yeah, great question. We were not able to do this type of quantification because in vitro, when we did the flow cytometry analysis, almost all the BEN3 cells were labeled with the vesicles. And when we did the analysis on the zebrafish model, we were not able to really quantify it. But for sure, this is not the majority of the vesicles which are crossing through the BBB. The BBB is still playing a role of filtration and protection of the brain. So we have a certain amount of the vesicles which are crossing through the BBB. But among all the vesicles which are interacting with the endothelial cells, we have vesicles which are degraded or modified or changed during the process of the crossing. So I don't know the amount. Maybe it's like 10%, I could say, but I don't know. Maybe Julien, you have a better answer about that.
Oh, don't say about that. It depends on the source of EVs, actually. You can have very low percentage of passage. In my end, for cancer-derived EVs, we can have very, very low rates. So it could depend on the pathology you are focusing on the population of EVs you are focusing on. So it depends. Randy Stute proposed a very long question, so I suggested him to turn on his mic just to ask him himself. So hello, Randy.
Hello. I was just I asked the same question that you asked first initially there. But the follow-up was if you see evidence that tight junctions are being disrupted due to the inflammation, and then on through to once they get past the endothelial cells, how do you suppose that are they getting taken up and then transcytosed again by the astrocytes, or are they how are they getting around the end feet, which entirely cover the vasculature in adult mouse and human brain?
Yeah, thank you. And I also have the same question because for sure that the in vitro model, we didn't have, like, we just have a monolayer. So it was very easy for the vesicles to go through the endothelial cells. For the zebrafish model, the zebrafish has, which is called glial-like cells, because there are some astrocytes feet connected to the endothelial cells. So we have some astrocyte and feet on the BBB. So it mimic a little bit more the BBB model compared to the in vitro model. So it like helps a little bit to have a more complex model, but for sure is not enough. So I only can suggest that in the human brain, for example, or the mouse brain, you have lower amount of the vesicles going through the BBB and reaching the brain. Also, another possibility is that depending on the composition, the peripheral EVs may modify the activity of the BBB, either the astrocytes, the endothelial cells, or the platelet, for example, and those cells may release, for example, toxic EVs or more pro-inflammatory EVs that could then affect the brain osmosis. But this is why I would like to continue to work on the passage of vesicles through the BBB, because we still have to understand what is really going on in more complex models, and especially in the human brain.
Right, so let's pursue with two questions. I'm just melting two questions from Mingling Liu and Pingping Chen. So which pore size of transfer did you use, and which kind of cell type did you use? And linked to a third one, why BN3 cells and not BN5 cell line?
Okay, so the first one was the size of the transfer?
Yeah.
So I have to remember because it's been like 2-3 years now, but I think it was 1 micrometer, 0.1 micrometers. I have to remember, I have to check in the, for sure it's in the paper, in the methodological paper, yeah.
I would say that it was close to 1 micron because endothelial cells are very, very small. So the pore size don't need to be so much high because the stability of the endothelial cells won't be so good. So you're true with 1 micron, I think.
Yeah, I think it was. Thank you. Like, sorry, like it was a long time ago, so I don't remember very clearly.
So yes, so there is some BN3 in the system. Which other kind of endothelial cells did you use, and why did you choose BN3 and not BN5 cell?
Good question. Well, we had this cell line in the lab, so it was easier for us to work with it, and also it was well characterized by different lab and ours. So this is why we used it, but I don't really know very well the BN3/5, but yeah. So sorry, it's like it was easier for us to work with the BN3.
The BN5 is not the classical cell line for mimicking the blood-brain barrier, actually, but yeah, there is some new paper showing that the BN5 could be also of interest. But yeah, so that was a nice question from Smentor, so he will recognize himself. Another quick question, probably. What do you think about the EVs from brain cells? Would they also contribute to the mechanisms of microglial activation where they interact with the EVs that cross the BBB?
Yeah, totally agree with that. Here, when we did the analysis of cytokines released by microglial cells or A-beta to released by neuronal cells, we did it like on different models. So we were working on either microglial cells or neuronal cells that were not pulled together. But for sure, in the brain condition, I think that all the cell lines all the cell types are interacting with each other, and vesicles released by macroglial cells will modify the activity on neuronal cells, and in another way, EVs from neurons will modify microglia or astrocytes, and they also can modify the integrity of the BBB. Different studies have previously observed that brain-derived EVs, depending on their composition, if there is a pathology such as AD or other, can participate to the pathogenesis of the disease.
Smart question with a smart answer. So thank you, Smart, for this question. The next one from Wrecking. The question was about the injection of your EVs. The injection was done directly into the hippocampus. Is there a particular reason to choose to inject the EVs in this brain region rather than that into the blood circulation?
Yeah, thank you for that. Indeed, we could have done the injection in the blood circulation. We had one main issues in our experiment is the samples of the vesicles, because we were working with patients, AD patients, ADLI patients, and we didn't have enough samples at the time to do the injection, to do enough isolation of EVs to be able to do injection in the blood. Because for sure, when you inject in the blood, you have a dilution of the vesicles in the blood circulation. And so you have a lower amount of the vesicle reaching the brain. Others have demonstrated that EVs injected in the blood circulation of mouse model can reach the brain. So here we decided to just focus on the hippocampus because we know that through the BBB, but also the CSF, EVs can reach the periphery to the brain through the hippocampus. And also the hippocampus is a very important area for memory and is heavily alterated by during Alzheimer's pathogenesis. So this is why we focus on hippocampus. But for sure, it would be very nice to see how the EVs from EDOLI and AD patients cross the BBB in a complex model, such as a mouse model.
Another simple question dedicated to the dose of EVs you used. Was the BBB penetration in the in vitro culture is dose dependent, and how did you determine the dose?
Yeah, good question. So we indeed had a different concentration of the vesicles depending on the patients and the health state. So here we use the same concentration for all samples or vesicles for both in vitro and in vivo experiments. So we used 1 per 10 particle per ml because in physiological conditions, it has been proposed to be the concentration of the vesicles in the blood circulation. So we base all our results, all our experiments on the concentration to then be able to compare between the groups. But in another way, which is interesting, that we have a difference of concentration of the vesicles between the patients, and especially for Alzheimer's patient, we have an increase. So maybe if we increase the concentration of the vesicles, we could have a more important effect on the BBB integrity, for example, because we observe a decrease of the tear level, but not of the integrity of the endothelial cells. So maybe if we increase the concentration of the vesicle, we could have a higher impact, or also a higher crossing through the BBB and reaching through the brain.
You discussed that during your presentation, but it's clearly an important question to address. With only lipophilic dye evidence, how can you really be convinced that EV are crossing the blood-brain barrier?
Yeah, I know that lipophilic dye are not very well, are not perfect, if I can say. But we had different controls during the labeling and after the labeling to be sure that we have EVs labeled and not, for example, micelles or lipid contaminants. And then when we did the experiments, we were able to, so there are two different things. We were able to see how the vesicles were interacting with the cells, and we compared also our results to other publications, and it was good to see that we had similar results or similar interactions between the vesicles and the cells, which was a good way to see, okay, we have here labeled vesicles and no contaminants. And another way, when we did the analysis, for example, in the first article of the IL-6 released by microglial cells, and then we used the BBB model transfer also, we still were able to have this effect. So it is a good way to see that, okay, we still have a passage of the vesicles through the BBB and then an impact on the microglia activity.
Yeah, it's sometimes complicated to manage to use fluorescent probes, and somehow it's also complicated to manage to create a tagged protein derived EVs, because somehow it could also interfere with interaction with the receptor. So that's also a way to answer one of the questions. It's a checkbox as well. Another one was dedicated to the mechanisms of absorption of EVs. Some of the EVs are not absorbed, some are, and other even cross the endothelial layer. Do you have any idea of the mechanisms responsible of this difference?
So we didn't study the mechanism, so I can't say. I mean, I know that it's active because at 4 degrees we don't have any interaction. So for sure it's an active mechanism. I can't say which one is the most prevalent, for example. But different articles have shown that depending on the EV's composition, sometimes from the same cell source, but depending on their composition, they can interact differently with the cell's receptor. So I think that we may have different interaction depending on the taker. And also because we're working with samples from the blood, we have a lot of source and origin, even if some are more important than others. So it's also very interesting it would be interesting to understand better how it's interacting, but I can't have a very well specific response for that.
Thank you, Sao, for your answer. And thank you for your question as well, Savio. Another one, if your EVs from age and middle-age AD subject both induce microglial activation with different characteristics, how do you interpret the significant difference in their quantity measured in the circulation?
Sorry, I didn't have the last one. How
do you interpret the significant difference in their quantity measured in the circulation?
It's a good question. I think that the thing is that EVs from EDOLI or AD patient may have a different impact. My opinion is that in healthy aging, EVs may contribute to senescence related to aging. And so it could contribute both in the periphery, because we have seen that release of cytokines from monocytes and CD4+ cells, and also in the brain, if they are able to indeed cross the brain. So probably EVs from EDOLI patients participate to senescence, while EVs from Alzheimer's could mostly participate to inflammatory reaction associated to Alzheimer's. For the concentration, I think that also we have an important role here in the constant release of EVs, a chronic release of EVs in the blood circulation. And so the cells, the human cell, peripheral human cells, or the brain, the BBB are chronically, interacting with this vesicle. So even if we have a lower concentration, maybe we could have a long-term effect on this function.
And probably the last question, because time is just running out. Which factors are more critical for EV translocation across the BBB, vesicle size, or the presence of specific surface molecules?
Yeah, again, I can't have a specific answer for that. I'm very sorry, but I think again, it really depends on the cell of origin and the homeostasis state of the cells and the composition of the EVs. Like for sure, the external composition of the EVs is very important. Its composition with proteins, ligand, different receptor will interact with the cells, but also the recipient cells also will be important for this type of interaction.
Just a small comment from Jagdeep Sandhu's question. I asked this question at the beginning of the questioning part. Yeah, the multi-omic approach could be a good point to go in depth to understand which kind of protein, which kind of cytokines, or which kind of lipids could also be involved in such regulations. So yeah, it's a nice question. So thank you to ask it again. So thanks a lot, Hermine, again for this nice presentation. Thank you everyone for attending this EV Club, such a great EV Club, and hope of seeing you as numerous as possible. Don't forget to follow the registered conference in the YouTube channel. So it's my closing remarks. I don't know, Ken, if you have some others to add, if you are here.
Thank you so much again for the invitation, and thank you everyone for being here and for all the questions. It's very interesting to have a feedback, so I will keep it in mind and continue to think about the next steps. Thank you so much.
It's just the beginning of a very long story, I hope. It's the best I can hope for you.
Thank you.
Thank you, everyone.
Thank you.